When you're preparing to collect a mushroom, there's several important features that I think you should be aware of. One of which is a little collection data sheet that we often use in a field, or a field sheet, in which we have the date, the location where the mushroom is being collected. If it happens to be on some type of plant, the identification of the plant that it's growing on, like an oak log or a pine log or something along those lines. Habitat is often important. For example, we're up on a kind of a dry ridge top here. If we were down along a stream, that would be a type of habitat to involve. And any field features that might be noticeable. For example, if you were to pick the mushroom and it changed colors, or if you cut it and it changed colors, or if there were milk-like substances coming out upon breaking or cutting. These are all features you should write down as they occur, because oftentimes the color will fade, the milk-like substance won't be exuded if you take it back after a few hours and try to get the same reaction. The color reaction may not be the same as well. In addition, when you are collecting, for example, this particular mushroom here, be sure to get the entire mushroom. In this particular case, we want to carefully remove the debris from the top. In the case of this is pine needles. But more importantly is to make sure that you get well underneath the mushroom to make sure you get all of the structures of the mushroom. Make sure you don't leave any behind, because as it turns out, a lot of these structures are necessary for identification. And if one is considering eating these upon collecting, and identification is very important, to say the least. On this particular case, we're dealing with a species of the genus Amanita. It has an annulus which oftentimes won't appear that commonly on the side for certain species, or on the stem, because it breaks apart and may end up hanging on the edge here. This one here has more or less collapsed towards the base. This has a very definite bulbous base to it, but it doesn't have a structure like a lot of the Amanitas had, called a volva, or a cup-like structure at the bottom. This is the structure that is often left in the soil if not collected properly. This particular case is confounded a little bit by the soil particles on the top, but if you look carefully, there are small patches of whitish fungal tissue left on the top, and these are also important. I often cut these in half in the field to see if there are any color changes inside or outside. And in this particular instance, and one should wait probably a minute or so to see if there is any color change. There basically is no color change in this particular specimen. Now, for continuation of collecting, I prefer to use, at least for the fleshy fungi, wax paper. And I'll include then the information or field note so that it doesn't get lost. I would recommend highly that you don't rely on your memory. If you write it down, then chances are pretty good you're going to be able to have that information when you get back. And then simply twist the edges like this. And this provides some very good air exchange, so the particular fungus, in this case this mushroom, will not dry out or become too hot during transport. It does provide a certain amount of protection from being squashed. And then place it in your collecting basket, such as that, and then you can be on your way.